If DNA is the instruction manual of your body, then proteins are the final product. They are the little molecular machines that make you you. They have many different jobs, which we'll talk about here in a little bit. At the molecular level, they look somewhat similar to DNA, but all molecules that make up living organisms have these similarities. They are made up of four basic atoms. So we've got carbon, hydrogen, nitrogen, and oxygen. And those four molecules make up much more than 90% of all of the living organisms. And amino acids or proteins are no exception. Now, we do have other molecules. There's iron and sulfur and potassium. My mind is going blank, but all these other ones that are critical also, and they are important, but they're found in much smaller amounts. Now, for proteins, which are made up of amino acids, we have these chains that are linked together. And this is a diagram showing you the basic amino acid structure. I don't expect you to memorize it. You don't need to know exactly how it works. But what you do need to know is that there is this nitrogen and carbon kind of backbone here, and that we keep adding one after another these little molecules polymerizing and making a protein. And each of these individual molecules is called an amino acid. Now, the R down here is a functional group. And if you've taken organic chemistry or maybe even other chemistry classes, you know a lot more about this. And so we're not going to get into, again, all the fine molecular details. But this R or functional group is what makes each amino acid different. It gives it its own individual unique properties. And we've got a diagram of all of the different functional groups here. And there are 20 different amino acids that make up living organisms. So, just like we have four basic nucleic acids that make up DNA, we have 20 different amino acids. Okay? And this polymerization, or this stringing together of molecules, is formed with what we call a peptide bond. And this is a covalent bond, so one of the strong type of molecular bonds. And we can link them together over and over and over again. Now, this process of polymerization, of building proteins, is done at the ribosomes. Okay, and so ribosomes are these little mechanisms, and they themselves are genes, but they're genes that are never translated into proteins themselves. They serve their final function as RNA. And they have this three-dimensional shape that the mRNA fits into. The tRNAs can come in, and we can polymerize and build these strings of amino acids. And then those amino acid strings, the proteins themselves, fold. Sometimes there's some post-translational processing that needs to occur, but many times, just given the correct cellular conditions, the stable homeostasis, salt conditions, pH conditions that are present in cells, these strings of amino acids will naturally fold up into their three-dimensional shape, and we get our final product. So that is the basic molecular structure of proteins. And again, that's much more important for chemistry, for biochemistry, but I do want you to understand it at that basic level. And just like DNA, we don't have a ton of variability. At the molecular level, we have four main atoms, right? Those atoms string together in these large molecules, and there are only 20 basic amino acids. So many more than there are at the nucleotide level, but still a fairly simple starting point. And by putting those 20 amino acids together in different combinations, we can get essentially, because there is different length, proteins are not a fixed length, and we can string these amino acids together in any combination, we can get essentially infinite numbers of combinations. And so we have tens of thousands of different proteins, and each of those can have several varieties when we start looking at allelic variation. And those make up an individual organism. Now, there are four structures that we can evaluate when we're looking at proteins and how they function. So you need to know these. The first one is the primary structure, which is simply the order of the amino acids. And sometimes you'll see amino acids with three-letter abbreviations in MEGA, which is the program we'll be using primarily for the worksheets, but to illustrate a few other things. In that, you have one-letter abbreviations that show us this primary structure of the protein. Okay, I like to think of it, I don't know if you guys ever played with pop beads or like beads on a string, but when I was a young father, my kids had these toys we called pop beads. And pop beads had a little hole at one end and then this little knob sticking out at the other end. And so you could string them together, but only in one order. And so we build proteins the same way. We can't build them bidirectionally. They only go in one direction. And the amino acid, if you look at the basic backbone, is also directional. We have the amino group on one side, the carboxyl group on the other, and so the amino group can bond to the carboxyl group, making that peptide bond, and then we can build them all out. And then hanging off of these are those functional units. There is also, once we build the primary structure, that dictates and determines the secondary structure. And secondary structure are these small parts, not the entire protein itself, but small parts of the protein that form these three-dimensional shapes. And the two most common ones are the alpha helix, the beta pleated sheet. And these are not really critical. You don't need to memorize these, but just know that these small three-dimensional structures are like the individual parts or components of the final protein product. And so the final protein product we term tertiary, or the tertiary structure. And there are multiple ways to represent this. So you can see some of these secondary structures. Here we have two alpha helices. Here we have a beta pleated sheet going this way and a beta pleated sheet going the other way. So this is just a diagram. This is called the ribbon model, but a diagram to represent that three-dimensional shape. Sometimes you'll also see the entire molecular structure in the ball and stick model. This is often not very useful because it's just so busy. There's so much going on here. These molecules can get very, very long, and once they fold up, it's really hard. You can't even see the primary structure anymore. You can't even really make out secondary structures with this, but it gives scientists a very general idea. So more frequently, you'll see a ribbon model or perhaps even a space-filling model. Now, the final product for some proteins goes even beyond the tertiary. So we transcribe and translate one protein. It folds into its final three-dimensional shape. That's the tertiary structure. And then often another unit, sometimes the exact same unit, so they're doubled up in a dimer or trimer or tetramer in 4, but oftentimes closely related or even very distantly related proteins will get put together to form the final protein product. So here we have 4 different tertiary structures that all go together to form the final protein product. So hemoglobin, for instance, is a combination of different tertiary structures that all make the final working protein. And many proteins, but not all proteins are like that. Some proteins are functional just on their own, that we don't have a quaternary structure, but many of them do have this final step where we have multiple little protein products, sometimes all from the same gene, sometimes from multiple genes that all get packaged together to make the final working protein. Now, what do proteins do? Well, we've already given you this big picture, and the short answer is basically everything except for being the instruction manual and energy storage, although some proteins actually are energy storage, but the main energy storage, at least long-term energy storage in your cells, is a lipid. But proteins do a little bit of energy storage. They do everything else except for being the instruction manual for the cells. So let's just look at a few examples. This list is not exhaustive. We're not going to cover every single possible function of proteins because it's vast, but we'll cover much of it, the main ones here. So one really important one, this is important in immunity, there's been a lot of talk of it lately, is antibodies. An antibody, and this is a three-dimensional structure showing what an antibody looks like. An antibody is a protein that has a variable end that binds to an epitope or a target on some sort of invader. And then it has a conserved end that interacts with your immune system and basically serves as a red flag for that invader. So your immune system can target it, attack it, and keep you healthy. So right now, right, of course, we're worried about antibodies for coronavirus, right? And the idea is that the way the mammalian immune system works is that once we make antibodies for a product, for a target, like coronavirus, we often then will get some sort of acquired immunity, often very long-term and strong immunity, so we can't get reinfected, at least not by that exact same invader. And we're hoping, there's still some research, but it looks somewhat promising, that at least there'll be some long-term sustained immunity against coronavirus. And you can generate antibodies either by being infected with a bacteria or a virus and then having to fight it off, and then your body, your memory cells, keep producing that antibody. Or you can gain them through an immunization. We inject something that mimics the original bacteria or virus but doesn't make you sick, and then your body generates antibodies to it, and then hopefully, that's the point of a vaccine, you are then immune. So really critical for our immune systems. Proteins can also be contractile. This is a diagram showing actin and myosin, and we're zoomed out a little bit. They've shown these basic shapes of the entire units. But actin and myosin work together a little bit like a ratchet, if you've ever used one of those tie-down ratchets, but they can be ratcheted forward and then relax and then expand. So we can contract and then expand. And this all happens very, very rapidly over milliseconds, so we can rapidly contract our muscles. The vast majority of our muscles are made up of actin and myosin. And these cells are also these molecules are also used in other places, like in cells as part of the cytoskeleton and for other structural elements. But this is what makes animals able to move and react to their environment. Many proteins serve as messengers. Now, there are other molecules that can serve as messengers also. But for instance, insulin is a protein that signals, it serves a message. It's created in our pancreas, released into the blood, and then it goes to the cells and tells the cells that they need to take up sugar and store sugar. And then there's another protein called glycogen that does the opposite, right? So, sorry, let's reverse that. Insulin is the signal to release stored energy in the form of sugar, and so that's what regulates. It's part of the cycle that regulates your blood sugar. So glycogen and insulin together work to maintain your blood sugar levels. Okay, so this serves as a messenger from one part of the body to another one. And there are many, many other proteins. And again, you don't need to memorize any of these or look at the structure. Here we have our little stick model with the space-filling model above it for insulin. Okay, but it's a messenger, one of many protein messengers. Many proteins are structured. This is a diagram of collagen, which is kind of like an elastic protein found in high volumes in your skin, especially when you're young. And so when you pinch your skin to make it out of shape, and then it bounces back, that's largely collagen. There are other proteins and structures there, but largely collagen that helps it to bounce back. So it maintains that important structural. As I mentioned, some proteins are for energy storage. Casein is one of those, which is an important storage molecule in mammalian milk. Now, for really long-term storage, lipids are really what's used. And lipids are a different class of molecules from proteins or amino acids. They're different. And just as a review, I guess maybe it's worth mentioning here, there are 4 basic macromolecules that make up all living organisms. Right? The vast majority of living organisms are made up of nucleic acids, which we talked about already, RNA and DNA. Amino acids, which we're doing now, which are peptides, right? Proteins. And then we also have lipids and, of course, carbohydrates. Okay, so all of them are made up primarily of nitrogen, carbon, oxygen, and hydrogen. However, they each have their own specific function. Of all of them, proteins is the most flexible, the widest range of functions. And so there is storage. Maybe you think of it as medium range storage of energy, and casein is just one example of that. Proteins are also very, very important transport molecules, and these can be either transport long distances, which is kind of like a combined messenger transport function, or very commonly, they serve as the transport mechanism from the outside of the cell into the cell, right? So we have these channel molecules that either help with facilitated diffusion or active transport of molecules into the cell. Hemoglobin is one example of a transport molecule that goes over long, long distances in the body. So here we have 4 oxygen molecules bound to the 4 different subunits of hemoglobin, right? This is the quaternary structure. So transport molecules are a very, very important function of proteins. Proteins can also be enzymes. An enzyme is some protein that catalyzes a reaction. It causes a chemical change. And it does this by lowering the activation energy so that we can not have to dramatically raise or lower the temperature of our cells. Our cells can stay one temperature and one pH and all the other great balanced values that maintain homeostasis in our cells. And we can still get things done because of these little catalytic molecules, these enzymes. This is a diagram of a very complex enzyme called ATP synthase that helps to add the third phosphate group onto ADP to make ATP, which is the short-term storage molecule, right? So that is a very, very critical function, but there are many, many, many other enzymatic functions. And enzymes are adapted and evolved to be most efficient at cellular conditions, depending on where the enzyme is. Most of them are in cells where we have this very stable cellular conditions. Some of them are in the blood, and so we have slightly different conditions in the blood than inside the cells. And others in some organisms are made to work externally, like maybe on the surface of the skin. So they're adapted to slightly different conditions, but most of them are adapted to these cellular conditions. And if we have an organism that lives in different conditions, like a hot springs bacterium, then their enzymes are adapted to be most efficient and work best at those specific conditions where they live. And that's part of the natural selection evolutionary process. Proteins are also critical for gene expression. And this is kind of a little bit of a circular reasoning or a catch-22. And this evolved very, very early on, but now is a very tight, integrated, interlocking system where there are certain proteins, primarily called transcription factors, that bind to DNA and help to activate and cause transcription of that DNA into an mRNA. That mRNA then gets translated into protein. So in all living organisms today, we need proteins to make more proteins, right? So again, you can see the integral vital function that these proteins play. So again, that's not an exhaustive list, but it's a very good long list and gives you an idea of some of the major jobs of proteins. Proteins are critical for proper cellular function. Shape of the proteins is critical for proper protein function. And so this is why we very, very often display proteins in this tertiary and quaternary structure, because that is the functional working unit.